All right, everyone. We are about to start. Can you hear me okay? Can you see some thumbs up or something? Yes, nice. Great. People are still coming in. I am going to start introducing today's webinar at UAC, our second one of this new semester, and our second one also highlighting the work of the new PIs that are coming now at UAC. And today we have a talk by one of these new PIs, Professor Christel Bergström from the Department of Pharmacy here at Uppsala University. And she's going to talk to us about a very interesting new project looking into how we can improve the delivery, the oral delivery of antibiotics. So she's going to talk to us about their methods, the way that they're setting up the project, and what are they hoping to get with this work. I will now stop sharing my screen and leave the floor to Christel to hear her story. Thank you so much, Christel.
Thanks a lot for the invite to present our research here today. We are super excited to join UAC with this project and learn from all the great PIs that are on board. Let's see if I can get my presentation up and running. That should be, is it in the right mode?
Yes, yeah, I see it correctly. Yeah.
Great. So what I'm planning to do here today for the next 30 minutes or so is to simply describe what we are working with in my group here at the BMC at Uppsala University. And I will talk you through a little bit about different methodology that we have developed and that we can make use of to better understand the performance of dosage forms. And in this particular project that we then dock into the UAC, we will focus on, of course, a better delivery of antibiotics, focusing on an improved intestinal absorption of such antibiotics to also reduce the side effects of the provided antibiotics. So I added here to my title that we have a focus on dosage form design. So the team that I lead at Uppsala is about 25 people nowadays. We are a highly international and interdisciplinary team with about 12 nationalities. We also are funded by different sources, and I would in particular like to acknowledge the European Research Council that has funded 3 projects so far within my group. I will start to talk a little bit about the molecular perspective and molecular perspectives on drug absorption. So what we really try to do is to link the molecule with the dosage form and the physiology of the patient to improve the drug delivery. So for me, it's always about setting the molecule in the center of the research and try to understand how that compound can inform us about how it will be processed by the body. And that can be done by linking different physicochemical properties that you can actually calculate directly from the molecular structure to then physiological properties such as the solubility in the gastrointestinal tract, or the membrane permeability and potential transporter interactions. And in our case, we are in particular interested in the intestinal epithelia, intestinal membrane permeability and transporter interactions occurring at that level. So with knowledge of these molecular descriptors, one can actually predict these different properties. And for antibiotics, we know that they classically have been poorly permeable, so they have difficulties in crossing across the cells or get into the cells. But also recently, we have a lot of issues with solubility around also antibiotics. If we have difficulties with membrane permeability and in particular then also with solubility, one can make use of different kind of formulation strategies. I will focus on today's talk on what we can do with lipid-based formulations and then also absorption enhancers to improve both the solubility and the membrane permeability. So solubility, if we start with solubility, about 70 to 90% of all drug discovery molecules that are in the drug discovery pipeline nowadays are expected to be poorly soluble. And that means when I talk about poorly soluble, it means that the compound cannot be dissolved in water, and in particular then not together with a glass of water that you would take with your potential medicine. And one can question how bad this is. So I typically show this case to people that don't know how bad it is with the solubility of compounds. So I here have a solubility competition between marble. So this is Venus de Milo to the left, and felodipine, which is a drug that is provided in a relatively low dose, 10 milligrams. It's used to treat hypertension. If we compare solubility of felodipine, which is sort of a quite representative compound for the discovery stage and what kind of compounds that we get in the hands of the formulation scientists. If we compare the solubility to marble, between felodipine and marble, it's actually so that the marble is 60 times higher in its solubility. So there are significant issues around solubility and trying to deliver a concentration that is high enough to the body so that we actually get the therapeutic effect. And when it then comes to the antibiotics, this is then followed by also typically a poor permeability across the membrane. So when it is linking the molecular structure with regard to solubility and how we then potentially later on can make use of dosage form design to improve this concentration gradient driving the absorption, we can make use of two properties to identify what the reasons for this low solubility is. So one is the melting point of the material, so looking into the crystalline structure and determining how easy or difficult it is for the molecule to dissociate from its crystal lattice. So when the melting point is greater than 200 for a compound, we know that that would significantly influence the resulting solubility. So the solid state itself will influence the solubility. And these are quite commonly nowadays described as brick dust molecules, trying to visualize that these compounds are as difficult as stone to dissolve in. The other property that may be limiting solubility, but actually were quite beneficial for the permeability, is lipophilicity. If lipophilicity is too high, then the compound do not want to interact with the water and cannot dissolve in the water. So if we have a partition coefficient between octanol and water, which is commonly used within medicinal chemistry as a descriptor of lipophilicity, if this factor is greater than 3, that indicates that we have a significant issue with getting this hydration of the molecule going. These are commonly called grease ball molecules, here just presented and visualized with some butter. So these compounds look quite differently from each other, so we can actually chemically see more or less on the structure if it's a grease ball or a brick dust. So grease ball molecules are relatively large in the small molecular space. So we are still talking about relatively small molecules here, so maybe up to 800 in molecular weight for those of you that are interested in chemistry. But they are also more flexible and have more rotatable bonds, and as said, more lipophilic than the brick dust molecules. So brick dust molecules are typically smaller, and they have quite large fractions of their surface, molecular surface, being rigid in different kind of cyclic systems. And this rigidity then enables these compounds to pack densely, come closely in the crystal lattice and pack densely, as compared to if we have a highly flexible molecule that can reform and have many different conformers, difficult then to interact closely in the crystalline lattice. Making use of molecular descriptors, one can then predict solubility in the intestinal fluid. This is just some data where we have made use of multivariate data analysis to predict solubility in simulated intestinal fluids. This is with a PLS technique, and it goes a few years back in time, where we had a relatively large data set, which we had experimentally determined in this simulated intestinal fluid and made use of as a training set for this model development. Then we used the trained model to predict new compounds, both an external test set just taken from reference literature, and then a test set that we got from AstraZeneca, who we collaborated with in this application. Here you can see the accuracy of such a prediction when we only make use of molecular descriptors. So the light green is the compounds that were trained on the model, and then we have the test sets in the darker green. It improves a little bit if you also include a commonly available experimental measurement, that's solubility in a buffer at pH 6.5, then mimicking the pH of the small intestine. But as said, actually the molecular descriptors themselves can provide information of the solubility. So this can then be combined with predictions of permeability. So what I show you here is a classification of compounds. In this case, more than 1,600 compounds are visualized here as these bubbles. And we go from green down to red. And if you're up in this corner, you have a compound that both has a good solubility in water and a high permeability across the intestinal wall. Whereas if we're down in this corner, we have compounds that suffer both from poor solubility and poor membrane permeability. If we put classical antibiotics on top of this, we can see that they are typically positioned in this space, so in the yellow-red space. Traditionally, as said, they used to be poorly permeable across the membrane, but also the emerging antibiotics are showing poor solubility. So here we would need to make use of different kind of formulation strategies to try to push these all the way up to this area where they are both soluble and permeable. If we look then into the environment that these formulations and drugs are exposed to after an oral intake of a medicine, it's a highly complex environment in the gastrointestinal tract, and it's also dynamic and changing over time. This is a graph then showing a part of the small intestine, the duodenum, where we have the bile duct coming up. So if we would be in this end of the graph, we would be in the stomach, and down in this end, we would be in the colon. When we move across the gastrointestinal tract, we have a pH gradient that the compound is exposed to. So in the stomach, it's a very acidic environment, and then moving up to become more and more towards the neutral stage. So 6.5, 6.8, something in the small intestine. In addition to that complexity in the change of the pH, we have bile secretion, and the bile secretion pattern between individuals is very different. Both the pattern when it secretes and the content, what is secreted, so the composition of this bile. And I guess that everyone listening in here today, some of us have had lunch, some of us will have lunch after, and dependent on what we have eaten or not eaten, the bile will be very different in the bile composition, and then the intestinal fluid composition will be very different in each and every one of us. This is important since these lipophilic compounds that are poorly soluble, but typically then better membrane permeable, they can be solubilized in these structures that are formed by the bile. So different kind of nano aggregates that are formed can then incorporate these highly lipophilic compounds. And then we also have the further complexity then coming from the food that we eat and lipids that are included in the food, or potentially lipids that we make use of to improve the delivery of the compound. So lipids that we include in the dosage form. So this is just a graph of what it looks like. This is aspirated, so duodenal fluids. So one goes down with a tube and you suck out a volume fraction of the fluid that is present in the duodenum. And this is a collection from 20 healthy volunteers. It's a work performed by Professor Augustins at KU Leuven in Belgium. And you can here see that indeed we differ a lot in between individuals, both in the fasted state, so that is before we eat anything, and in the fed state. We can see that it's not only the bile composition that is different, it's also the pH profile that is significantly different, going from relatively acidic in the small intestine to up to more into the basic side. This will then influence the solubility of the compound in these intestinal fluids, as well as the interactions with these colloidal structures or nanostructures will do. So just to show how big difference it can turn out to be then if we take a medicine. So this is Peridol. I have plotted or printed some of the physical chemical properties here if you're interested in physical chemistry. So this is a relatively ordinary drug molecule for the time being. It's about 500 in molecular weight. It is a lipophilic compound and it's slightly basic. So it can be ionized and be present in its cationic form in the gastrointestinal tract. From 10 healthy volunteers where this fluid was aspirated, we determined solubility. Or so not we, the group in KU Leuven determined solubility. And just by looking into the results here, we can see that we have a 20-fold difference in solubility between individuals for this particular drug. And of course, that significant difference in concentration that is reached in the intestine will then significantly influence the absorption and the amount that is reached the blood circulation to then potentially treat the disease that is targeted. So this is, if we lean back to what I described previously around the grease ball and rictus, this is a lipophilic compound. It's not significantly influenced by its melting point, and it's then classified as a grease ball molecule. If we look into details, so what will sort of limit and what will drive solubility, what is driving solubility is the ability for this compound to form intermolecular hydrogen bonds. And it's also good if those atoms that can do so, like nitrogen and oxygens, are distributed evenly out across the molecule, so that we have several anchor points for interactions with water, so to keep it in solution. However, if we compare this now to permeability, membrane permeability, it is in principle the other way around. So if we have lots of intermolecular hydrogen bonds to water, that means that we have lots of polar groups. Then the compound has significant difficulties in crossing through the lipid bilayer of the cells. So it's not easy to drive the absorption, because if you manipulate or have a compound where you have a good solubility, typically that is followed by a poor permeability. But as said, we also have now the compounds that both show a poor solubility and poor permeability. But we can make use of formulation approaches to solve both these. And one formulation approach that is currently used to solve both the issue with solubility and the issue with permeability is lipid-based formulations. So I will just go through very briefly what lipid-based formulations are, but one can view them as you provide your compound together with oils or oily mixtures, sometimes relatively complex oil mixtures, but similarly to capsules with the omega-3 fish oil, these would be capsules with similar kind of oil, but also with a dissolved drug in that capsule. So the benefits with this system is that if you deliver it with the oil, these lipophilic compounds, they really thrive in the oil. So you can provide it in its dissolved state. So that makes it freely available. As soon as it's released out to the intestinal fluid, it will be available for absorption. So it doesn't have to dissolve after oral intake. Then these excipients, or the lipids and different surfactants that may be used in these formulations, they inherently increase the intestinal solubility by what is called solubilization. So they form also these different kind of nanostructures in the when it's dispersed into the intestinal fluid, and these can then maintain or keep the drug solubilized in the intestinal fluid. So this enables delivery of very lipophilic drugs, but it also enables us to encapsulate and potentially present a poorly permeable compound closer to the membrane, or even make use of different kind of pathways that are triggered by the presence of the lipids. I will come back to that by the end of it. So if we provide a lipid-based formulation, then the lipids are digested when it hits into the intestinal fluid. It's digested by different kind of lipases that are secreted through the pancreatic duct. You can then study this in the laboratory, and the common way to do that is to make use of a technique called lipolysis. What you do there is that you, in a closed beaker, you add a simulated intestinal fluid together with your formulation, where you also have then your drug inserted, and enzymes that can start to digest these lipids. So what you measure is the extent of digestion, and as this digestion occurs, then we have a change in these solubilization capacity, or these structures that are formed. So there is a high risk that when these lipids are digested, the compound will not be able to stay in solution any longer and precipitate out in its solid form. If that happens, then the whole nice work that we've done with the formulation strategy is gone. So, and the benefits that we were expecting from the formulation is not reached. So it's very important to understand how this digestion potentially may influence the resulting solubilization capacity and the resulting concentration gradient that can drive absorption across the intestine. We can also then study where is the drug after this digestion. So has it precipitated? Do we have it in its solid form? Is it still sitting in the undigested oil droplets, or is it in the water phase available for absorption? However, when you do this in the commonly used lipolysis methodology, you don't have any absorption compartment present. So it's a closed beaker without taking into consideration that actually both the drug, the lipid, and some water is absorbed as the digestion is occurring. And that means that it is not a good translation to the in vivo situation. So what I show here is what we can measure in vitro, so in the lab, on the lab bench. We would, for 3 different lipid-based formulation, predict one to be much worse than 2 others. But when we do an animal study, in this case in pigs, we can see that they actually perform equally well. So all 3 performed or gave the same concentration in the blood plasma. So we have tackled this by developing a new device that we call the ENA device, and the ENA stands here for the enabling absorption, where we have combined this lipolysis in an upper chamber with an absorptive membrane composed of, in this case, CACO2 cells, that is then mimicking the intestinal wall, and then added a receiver chamber that is mimicking the blood. So what we now then can do is to measure digestion similarly as before in the upper chamber, and we can measure the drug distribution, so how much of the drug is available for absorption. But importantly, we can also measure how much is actually transferred across this cellular barrier, and then predict how much would reach the systemic circulation. So not only looking into the processing in the gastrointestinal tract, but also looking into what that processing do to the flux over the intestinal wall. When we have this mass transfer, we can relate that back to in vivo data. So in this case, we have shown that this is an in vivo predictive tool, both when it comes to data that is generated in pigs and data that are generated in dogs. In combination with this sort of experimental development of new technologies in our laboratory, we are also focusing quite a lot on different computational strategies. So I showed you early that you can make use of different kind of machine learning techniques to predict solubility and also permeability in these very complex environments. But you may also make use of what we have established and call a virtual intestine, where you can look both into the performance of the drug itself. So where does this drug go? To what extent is the drug solubilized in these different nano aggregates that are formed in the intestine? But you can also look into the formulation perspective and what the formulation do to the delivery. For the virtual intestine that we have developed, we have made use of molecular dynamic simulations. And with these molecular dynamic simulations, you can study a lot of different kind of molecular interactions. So both between the drug itself and these different kind of structures, but you can also look into phase changes of, for instance, the formulation that you are interested in. You can do this at different resolution scales dependent on the process that you are interested in looking into. And we have decided in our work to do most of our studies with what is called a coarse-grained MD simulation methodology. By using coarse-grained, we reduce the resolution of the molecule a bit, so we don't get all the detailed atomistic interactions from these kind of simulations. So this is visualized here with that a number of atoms from this molecule is now lumped and presented as a bead. So this bead should now carry the information and physical chemical description of all 4 atoms sitting in this part of the molecule. So by reducing the resolution and the degree of freedom for the molecule, we save a lot of time. So I said here that we save a lot, at least 10-fold faster, or we are at least 10-fold faster when we make use of this technique. So either we can run the same simulation much quicker, so we can do many more simulations, or we can do more extensive simulations. So either in time or in size of the environment that we are simulating. And by increasing time, we can capture the process kinetics better. And by increasing size, we can capture a larger fraction of, for instance, than the intestinal compartment in our simulations. We are still in a very short time frame and small volumes, but by doing this change, we actually have the capacity to capture phase changes and aggregation and so forth. So what we did when we established the virtual intestine was that we collaborated with Professor Patricia Agustins at KU Leuven. The professor that I showed had done all the sampling to look into the inter-individual variability in our intestinal fluids. So he provided us with the exact compositions of these 20 healthy volunteers. And based on that, we selected 5 individuals for which we put in the exact same compositions in our MD simulations in the computational toolbox and simulated these fluids. So in the end, we have 5 different intestinal fluids, and you can see them here as the final snapshot of these simulations. So what it looks like in these fluids. We have developed and used during SciFest virtual reality and 3D goggles for kids, and they were super excited to see all these bile salt that is in gray here, shuttling back and forth between these nanostructures and how these nanostructures also change over time. But what we clearly can see here is that we have very different compositions, and that means that we also have different capacity to dissolve these poorly soluble compounds. So here, for instance, we have not that many nano aggregates as in this. We also see that these are more green than in this healthy volunteer. And that means that this composition is more based on lipids, phospholipids, whereas this person had more of the bile salt composition. And these are significantly different. So if we look into what they do to the micellar structure or the structure that is formed, these nano aggregates, we can see that we have a lipid core, and on the outside of that, the bile salt form a shell, so sitting sort of a protection or an intermediator between the water and this lipid-rich core. What this do to the solubility, we can see here. So this is also now a computed solubility experiment where we looked into the affinity between a drug molecule and these different kind of nano aggregates, these lipid core and bile salt aggregates. So here we have looked into 3 drug compounds. These 3 drug compounds are neutral, so they don't carry any charge. So we try to simplify the system a little bit in this case. And they are of different lipophilicity. So prednisolone is the least lipophilic and propofol is a very highly lipophilic compound. We would then expect that if these simulations have worked well, and if this is sort of a trustworthy way to do the virtual assessment, the propofol should be significantly more solubilized in these fluids than the prednisolone. We would also expect that we should have a high variability in the solubility value for the propofol as compared to prednisolone. If we look into the upper graph here, this is interactions with one mice. So going back to the graph or the image I showed you before with only one such structure. And we can see indeed that this, in this case, the propofol has much greater affinity to bind to that structure than what the prednisolone has. We can also see that we have a much higher variability for propofol in the affinity to bind to these aggregates than what we have in the prednisolone. On the lower graph here, what we did was to do a volume fraction calculation. So this is more a representative image of what it would look in the complete intestinal fluid. But again, there we see that we get the same information back, that propofol would have significantly greater solubilization, but also a greater tendency to have a large variability in its solubility. So here it's sort of indicating that here we can be at risk with regard to having a variable concentration reaching to the bloodstream and then exerting its effect. We can also look then into permeability enhancers, and these may be of importance for those poorly permeable compounds, for instance, some of the antibiotics. So permeability enhancers that have been classically studied, they are based on fatty acids, and the fatty acids are also digestion products of these lipids that I've talked about previously. So actually what I show you now, we have studied it for permeability, from permeability aspects and to try to increase permeability, but these will also be present when we have digested a lipid or that we have taken in from food, or digested a lipid that comes with your dosage form. So what I have here is a nano-intestine, one can say. So we have a bilayer here that is enclosing the simulation. So this would be sort of the apical membrane of the enterocyte in the intestinal tube. And it's then enclosing the human intestinal fluid. So it's a nano-intestine that I show you here. In this, we have added these nano-aggregates from the human intestinal fluid that we simulated before, and also to that added those fatty acid components that are released during digestion. Here studied for the purpose of permeability enhancement. And the fatty acid is here denoted in red. So the red bead, if you follow the red bead, you see how this free fatty acid is distributed through the water phase into the lipid membrane. You can also see that when this is happening, you have here, it's only distributing into the membrane that is closest to the water phase. So we don't see any flipping of the lipid over to the other side of these leaflets. That is due to that it's a highly charged compound here, so it doesn't really want to do a flip in the lipid membrane, so it will not distribute through. What we can see is that we have a drainage, one can say more or less, of the free fatty acid over time. So starting with shuffling it through the water into the membrane, when that happens, a redistribution of the fatty acids from these nanoaggregates out to the water phase and then into the membrane. If we instead look into the same compound, but now completely neutral, so now we follow the green, this fatty acid does not now have any charge. We only get one large aggregate when we add this into the human intestinal fluid. So starting from the same point as before, with a lot of different aggregates, now all of them are lumped together. We don't see much of a shuffle of this free fatty acid via the water phase into the membrane, but suddenly when this aggregate is hitting the membrane, it's fully incorporated into the membrane. And what is also interesting here is this neutral lipid can distribute both, it can distribute through the lipid membrane by the flip-flop kinetics. If we now connect this to what we are going to do in our project at UAC, I will not talk that much about the project. I hope that we can come back and that we then can present you with some results of this very exciting project. But what we will do here is to focus on encapsulation of antibiotics into a lipid structure. In this case, we actually don't want these lipids to be digested. We want them to form nanoparticles that can come close to the intestinal epithelial and make use of endocytosis processes to deliver in an efficient way the antibiotics into the cells. By doing so, we would protect, that is at least our hypothesis, we would protect also the microbiome of the gastrointestinal tract so that we get less side effects. And we would increase the absorption so that we get less of the antibiotics out through the waste, via the wastewater into the ecosystem. So in this project, I'm the PI together with Dr. Madeleine Hubert, who is co-PI in the project. And since a month back, we have Shigo He, who has started as a PhD student. And we are super excited to embark on this project, where we will focus a lot on relatively sophisticated delivery strategies that we will characterize in different ways to get them as direct as possible and efficient as possible to be engulfed by the enterocytes in the intestinal lumen. So by that, I hope that I introduced you a bit to what we work with, what we can do, and hopefully maybe some of you could get some ideas on how to collaborate with us in the group also with your projects. And just as a summary for what I talked about, so from my perspective, I think it's important that you know your compound or the compounds that you're working with. So combining medicinal chemistry or making use of medicinal chemistry into the projects that in this case are also heavily involving physiology and delivery strategies, because they have the information that can help you to guide which formulations you use and which advanced drug delivery system that may be useful for your compound. We have developed new methods where we can simultaneously study digestion and permeation, and these are in vivo predictive and can inform then about the efficacy of the delivery system in fluxing over the compound over the intestinal wall. And then in the end, I showed you a little bit about the virtual intestine that we have developed, which we currently use to analyze and understand both solubility, digestion, and permeation. It can also then inform around how a formulation may be distributed or help to provide absorption. We have this available in the lab. We can make use of it already before you do any experiments to sort of reduce the workload and do smarter experiments. And just as a final note, as I said, we are super happy about being enrolled in the UAC, and we look very much forward, all three of us, to work together with you and to learn from all the very bright and competent scientists within the antibiotic field. So thanks a lot for listening in today, and please feel free to ask any questions you may have.
Thank you so much, Christel. We have here a question that someone wrote by Dietmar Witz is their name, I think. They're saying thank you for a nice presentation. And do you think the project can be translated from antibiotics to products, which will also come with low solubility and permeability?
Yeah, absolutely. I think, I mean, from a formulation perspective, we work with any type of compounds and any type of diseases. So I cannot see that there are any limitations on the compound side. There may be different tricks to play that you need to think of with regard to protection of molecules and compatibility with different kind of excipients that you may need to include. But definitely, I mean, lipids are highly interesting because they are typically also in the generally regarded as safe area. So we are exposed to lipids throughout our body. So it's an interesting concept here.
Interesting. I remind you that you all can either write in the chat or you can also raise your hands and you can speak out your questions to Christel. I have, well, first to say that you made me realize how incredible complex is taking a pill. The simple action of taking a pill and making it do what it's supposed to do, it has a lot of steps involved, and it's not as easy as, you know, just as you said, take a pill and take a glass of water, and it might not work for everything. So I thank you for, you know, highlighting this importance. I'm curious to know, in the process of developing new antibiotics or new drugs in general, at which stage do the researchers start thinking about this and working with real data to see how much is absorbed, how soluble or permeable they are?
So solubility and permeability is measured relatively early, and I mean, not even measured, predicted nowadays with the computational tools. I think, I mean, the computational predictions, different kind of machine learning's AI tools, they are absolutely good enough to indicate to you if you have a solubility issue or permeability issue. Also looking into different kind of metabolic patterns and so forth, toxicity, it's used early on in the projects. Then to set that in the context of what does it mean, I would say that comes far too late. So what does that mean for your project? What does that mean for you with regard to which kind of formulation may be needed for this particular compound? But because it's not necessarily so that you should do the same experiments for all, dependent on which kind of dosage form you're going for, or which kind of excipients that you need to have there. So if we are talking about lipid-based formulations, we have had quite interesting debate around if a compound is too lipophilic to be delivered, or if it's not lipophilic enough is the right term, not lipophilic enough to be delivered. Because the lipids, if you are using the lipids, you can really make use of relatively lipophilic compounds, and you don't need to close down projects or sort of deselect compounds because of that they are too lipophilic. That said, high lipophilicity may trigger toxicity, different toxicity profiles and different metabolic patterns and so forth. So there may be other reasons, but to weigh in all these aspects from the beginning, I think it's so you're informed early on in the discovery process around these different aspects, but then to do the synthesis and take all that information when you make your decision is not always done.
Mm-hmm. I understand. Thank you so much. Maria Selmer is asking if your work will also lead to better predictors in the future.
It depends on which predictors. So I think, we are currently, so we are continuing to explore around AI models for predictions of these highly complex or performance in highly complex environments, like in the intestinal fluid. I would say that the virtual intestine, I think, absolutely will provide us with better predictors. There, the limiting factor for the time being is, of course, sort of computational power. I said that I think that we can see a lot of very interesting things here in our simulations, but still we are simulating short time frames and relatively small volumes of the whole space. So. Mm-hmm. Not sure that was the right answer of the predictors, but I'm a big believer in that more data that we get in and the better use we make use of these AI tools, better predictors will be available.
Great. Mikhail Selin has his hand up. Mikhail, can you please pose your question?
Hi, Christel. Thanks a lot. There's clearly a lot going on at your end of BMC.
Yeah.
Yeah, I was curious about maybe an extension here. So you describe these formulations as a way of improving transport from the gut lumen and into the deeper tissues to sort of distribute antibiotics. But another key problem with failure of antibiotic therapy is that you have intracellular bacteria and they're in compartments that are hard to reach. Could you comment on how you think this may aid or not aid in reaching, let's say, bacteria that reside in vacuoles within cells, rather than just simply in the extracellular fluids elsewhere?
Yeah, that's an additional complexity, and I would say that for the time being, the systems that we are looking into are not as direct, so that they would target these bacteria. And if you look into the literature, there are many suggestions of what one can do. And you can study in vitro and potentially also in preclinical models, but typically very difficult to get it translated to a functional medicine in the end in the human, both with regard to reproducibility and with regard to the cost aspects of these treatments. So yeah, you can think of different ways to target once you have entered into, so that you sort of encapsulate your molecule in different layers, or that is activated or triggering different parts. But I would say that for the time being, what we are doing, we are not really into that in-depth direction of our formulations.
But would it be correct to say that if you with a formulation improve the sort of flux of antibiotics across, let's say, the epithelial layer, that should by definition also increase the concentrations within those cells as there is a transit through? So maybe you get that for free, sort of, at least in some tissues, or?
Yeah, so it would be sort of the passive effect of this formulation, that you would have a higher uptake, higher intracellular concentration of the particular drug that you're interested in. So sort of that would come with the game, so to speak, but not with an active targeting to that region within the cell for the time being.
Thanks.
But very, very interesting. That you start to spin out in many different. you get a lot of thoughts on interesting research projects that one could start.
Thanks a lot.
Nice. That's exactly where we are looking for as well, right? To be able to put people together, you know, that they might be interested in developing something new and something, you know, exciting and innovative. And I know Mikhail is working with also semi-in vivo models. So it's very cool, very nice answer. Any other questions? There's nothing on the, there is nothing here in the chat. Let me check. No hands up. And we are almost, let me put this in gallery view, and we're almost out of time. Yeah, it's now exactly 13:15. Thank you so much for the great timekeeping, everybody. It was great. Thank you so much, Christel, for your participation. It was great to learn the work that you are doing. I am more inclined now to say everything is much more complex than I already thought it would be, but that's just means that there is more more thrilling things to work on and to find solutions. And I really, really like that you are looking for innovative solutions, like creating new experimental systems that are as close as the in vivo situation and getting all the data that you can there. I thank you, all of you, for coming to this seminar. It was a success. We were almost 40 people this time as well. And I'm very happy to see a lot of the new PhD students also joining, getting on the gist of all the things that are important in AMR and antibiotics and antibiotic resistance. And Madeleine, thank you so much also for being here and your assistance in setting up this seminar. And I just hope to see you all back on the next webinar, which will be the first Friday of November. And with this, I conclude today's webinar. Thank you, everybody, and we keep in touch. Bye.
Thanks a lot.
Thank you. Bye-bye.
Thank you.